Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker on the question and answer section of the screen. After the presentation, we will begin the question and answer portion, and I will ask our speakers your questions. Your questions in the question and answer window are only visible to myself and our speakers. So thank you for joining us today. It's now my pleasure to introduce our speakers, Adrian Pappas and Chris Heger from Bio-Techne.
Thank you, Leah. Hi, everyone, and welcome to our webinar today titled Modernize Your Gene Therapy Analytics with Automated Tools from Bio-Techne. My name is Adrian Pappas, and I'm a commercial product manager for Protein Simple, a Bio-Techne brand. And I'd like to thank everyone for attending today, and I'm excited to welcome our featured speaker, Dr. Chris Heger. For today's webinar, I'll start off with a brief introduction, and then you'll hear from our speaker. And finally, we'll finish with a Q&A session to address your questions submitted through the presentation. This webinar is supported by Bio-Techne. Bio-Techne brings together the prestigious life science research brands of R&D Systems, Novus Biologicals, Tocris Bioscience, Protein Simple, Advanced Cell Diagnostics, and Exosome Diagnostics to provide the scientific research community with a comprehensive and world-class product portfolio of reagents, media, extracellular matrices, small molecules, assays, and instruments. In the last 18 months, we've developed and launched numerous products aimed at combating the COVID-19 pandemic, including SARS-CoV-2 proteins and antibodies, serology antibody tests, automated ELISA, automated Westerns, and automated CIEF and CESDS platforms to speed COVID-19 vaccine development workflows. And so now I have the pleasure of introducing our speaker for today, Dr. Chris Heger. Dr. Heger, Director of Application Science for the Analytical Solutions Division of Bio-Techne, received his doctoral degree in pharmacology from Cornell University and completed his postdoctoral training at the National Cancer Institute in Antibody Development, Purification, and Technology Evaluation. Chris joined Protein Simple in 2011, starting as a field application scientist and subsequently further building the East Coast support team as field manager. During these years, Chris developed a broad but deep understanding of the instrumentation and their application across diverse research areas. In 2016, Chris moved to California to lead the application science group, chartered with creating scientific collateral, fostering collaborations, training, and application development. Chris is an expert in immunoassays, capillary electrophoresis, protein and antibody purification, and chromatography.
Thank you, Adrian, for the introduction, and I would like to thank our attendees for joining me today to learn about how to modernize your gene therapy analytics with automated tools from Bio-Techne. Over the last few decades, there has been a tremendous evolution in therapeutics. There was a time where small molecules were the leading class, like Gleevec shown here. In more recent years, we've really seen an increase in biotherapeutics, and this really started with humanized monoclonal antibodies. Today, while there are still a number of antibodies coming to market to treat disease, pharmaceutical companies have realized and have begun to leverage new therapeutic modalities, including viruses, nanoparticles, and cells. The majority of current cell and gene therapies leverage two key viral vectors. For direct gene therapy via in vivo delivery, AAVs are the leading class. The other main therapy, called cell therapy, requires extracting cells from the patient and delivering targeted nucleases to the cells via physical, chemical, or viral methods. As shown in the schematic, lentivirus is used for transforming cells in ex vivo cell therapy. And after the cells have been programmed, they can be reintroduced into the patient, where they now can fight a tumor, for example. Importantly, both the AAV and lentivirus need to be produced in high amounts and of high quality in a process collectively called viral vector manufacturing. Viruses like AAVs are challenging to manufacture at scale. A high-level view of the viral vector manufacturing process is shown here. After gathering your starting materials, cells are grown and made to produce the virus. For these two steps, Bio-Techne offers a wide variety of research and GMP-grade reagents. After growing sufficient amounts of virus, the cells are lysed and the virus is liberated where it can be purified, and then formulated, and finally stored as finished product. For these 3 steps, we offer analytical tools and assays that are fast, robust, and don't consume a lot of the sample, which is quite important for this field. While I'm focusing on the viral vector manufacturing stage during this talk today, Bio-Techne can support the early research that goes into feeding this process. I wanted to briefly highlight our commitment to providing the highest quality reagents and consumables for the first 2 steps of the process. We recently opened a brand new GMP production facility in Minnesota and have begun producing GMP-grade interleukins for cell culture work. Many of our proteins can be formulated into our GMP-grade ProDots, which are convenient, single-use proteins lyophilized into sterile bags. And shown on the right is a table of the varying levels of quality we can provide for these products, along with the relevant testing and associated production requirements for each. Like any drug manufactured in the United States, viruses are subject to regulatory guidelines to ensure product safety and efficacy. These guidelines define a set of quality attributes that are to be characterized and tested not only at the end of the process, but throughout production. These quality attributes include identity, potency, purity, safety, and stability. In the column on the right is a list of the techniques used to address these attributes. And when I look at this list, I see a lot of tried and true but unevolved techniques. SDS Page has been around since the 1960s, analytical ultracentrifugation since the 1920s. Even Western blotting at 1979 is a pretty old technique. Bio-Techne, through its Protein Simple brand, has built on some of these foundational technologies and evolved them to a point that gives faster insights and ultimately a better view of your product. Let's take a high-level look at how Protein Simple can modernize the analytical tools being used to address the viral vector critical quality attributes. As we'll talk about today, our Maurice and Simple Western platforms can provide identity assays. For purity assays, our Simpleplex and Simple Western platforms can support host cell-related impurity testing, and our Maurice and Simple Western platform can provide information on capsid content. For potency, our Simpleplex and Simple Western platform can provide valuable information on physical and functional viral titer. And lastly, for stability, our MFI and Maurice platforms can provide information on aggregates as well as overall particle stability. Let's start with purity and identity. The USP Chapter 1047 defines analytical tests for both. For purity, analytical methods that separate, isolate, and specifically quantify the intended active product components determine product purity. And importantly, lot release testing for gene therapy products must include an identity test. This test clearly identifies the product and confirms the inadvertent substitution with another product has not occurred. Here at Protein Simple, we have 2 capillary electrophoresis or CE platforms to support identity and purity assays, and we can cover you from early to late stage. Our Simple Western platform is ideal for complex samples and features amplified detection, which is great for low abundance materials. As your material progresses and increases in purity, our Maurice platform can take over, providing industry-leading separations of your near pure or pure AAVs with direct detection. Together, these instruments provide robust analytical support for identity and purity assays using both size and charge separations. Simple Western is an ideal platform to support early-stage gene therapy analytical development. Featuring fast time to results with data in as little as 3 hours, we offer both size and charge-based immunoassays. The platform features quantitation with our built-in analytical software called Compass, and we're able to provide both absolute as well as relative protein quantitation. The Simple Western platform has throughput and flexibility. We can run up to 96 samples per run, as well as having multiplexing capabilities for even more data points per capillary, as well as a variety of platforms to fit your throughput needs. The Simple Western platform features excellent reproducibility both within a run and between runs. And lastly, and quite important to this field, is low sample volume. You can start with as little as 0.6 micrograms or 3 microliters per well to get picogram level sensitivity. Maurice is a dual mode instrument and provides the gold standard imaged CIF technology for charge heterogeneity analysis. It's used widely for monoclonal antibodies as well as fusion proteins, but also more recently, as we'll discuss today, for viruses. Like its predecessor, the ICE3, Maurice features direct detection with absorbance, but adds native fluorescence, as well as an optional blue fluorescence filter. Maurice features CESDS with direct detection for purity analysis of proteins. The platform leverages innovative cartridges that determine the mode of operation and makes the platform easy to use, as well as easy to switch between modes. We also offer CFR 21 Part 11 compliant software, as well as Empower control availability for deeper system integration. Across these two platforms, we have 3 CESDS or size-based assays to address identity and purity. The Simple Western immunoassay replaces the Western blot as we already discussed, and it is a great identity tool. It works well for capsid ratio measurement as well. Of these 3, it is also the most sensitive assay as summarized in the table below. The second assay on Simple Western is like an SDS page with stained proteins. In this case, our total protein assays leverage covalent attachment and subsequent label detection. This assay is great for purity estimation. Both Simple Western assays are well suited for early stage work where you have lower abundance samples and really fast answers are needed. The third assay is on Maurice, which is the best for capsid ratio and purity because of the direct detection with absorbance. While requiring more sample, the CESDS Plus assay on Maurice features the lowest RSDs. Let's now take a look at some data to see how these methods compare. The 3 assays provide similar data, which is great because it means the 2 platforms can support an AAV program from start to finish. Here's an example of AAV2 with all 3 assays. First, in the immunoassay, we are looking at 6 × 10⁹ GC per mil and detection of the viral proteins BP1, 2, and 3 in blue, orange, and purple respectively with the B1 antibody from Progen. We ran this on our high molecular weight kit to get the best resolution we could for the viral proteins. Shown in the middle is the 5x total protein assay on Simple Western in the 12:230 separation matrix with the same AAV2 sample, just diluted a little bit less at 5 × 10⁻11 GC per mil. And on the right is the Maurice CESDS Plus data. It clearly has the best resolution with better than baseline separation of the viral proteins. All 3 methods produce similar capsid ratios, with each having a distinct benefit of high sensitivity and specificity for the immunoassay, the ability to measure protein purity and estimate capsid ratio for the total protein assay, as well as with Maurice, and clearly the best resolution is achievable with Maurice as well. Let's now see what it looks like when we start evaluating the AAV process by combining the Simple Western assays together in the same run using our replex assay to look at some AAV-containing cell lysates. So the Simple Western platform is ideal for early stage work, and we're going to start by looking at some cell lysates directly after AAV production. We're going to produce quantitative data in under 5 hours with a replex assay, and in this case it's going to be an assay where we have an immunodetection of AAV proteins and then replex for 5x total protein to look at the impurities in the samples. So probe 1, identity, probe 2, purity. Look at 4 different AAV samples shown here. AAV1, AAV4, AAV7, and AAV9, all with the anti-VP123 antibody that I was showing earlier. You can see specific detection of AAV7 as well as AAV9 with no detection of AAV1 and 4. In addition, it appears that AAV7 and 9 have a different molecular weight expression for their VP3 protein. We can also estimate the content or capsid ratio for these samples, and shown here for AAV7 at 1 to 2 to 8, VP1 to 2 to 3, and for AAV9 a 1 to 1 to 11 ratio. Now looking at the replex, or the second probe of these same capillaries, you can actually see that they contain a lot of protein. This protein can of course also be measured, or the so-called total protein area shaded in gray, and these data can be used for subsequent normalization of the data. Once you've purified your AAV sample, Maurice CESDS will provide the best data for purity as well as capsid ratio by virtue of its direct detection. To show some more data with the CESDS platform, we ran the AAV2 sample same as before with 5 replicate injections and measured the percent peak area as well as the capsid ratio. Overall, the corrected percent peak area measurement was very precise, with all percent RSDs under 4.3. The observed capsid ratio for this sample was 7.6 to 1.3 to 1, with a low observed RSDs under 6.9% for the replicates. And as I had indicated on the previous slide, with Maurice, we were able to estimate this sample was 70% pure due to the presence of all the low molecular weight species. Both Simple Western and Maurice also feature isoelectric focusing, and this separation mode can be leveraged for some powerful data for AAVs. Shown in the first table are the 2 assays we're going to look at, identity and purity again, this time for capsid content. Both Simple Western as well as Maurice ICIF can be used for both identity and purity. The main difference here is for our identity assays, we're going to use a denatured approach, or break the AAV apart. And for the capsid content, we're going to keep the AAV intact. The assay details are listed below. For sample requirement, both assays use low amounts of AAV, below 10 to the 10th GC per mil. They're both quite reproducible with fast time to results and offer sensitive detections, as we'll get into. The capsid protein ICIF method is platform and can be used for identity as well as stability indication. We first assessed the applicability of the capsid protein IEF assay to a variety of AAV serotypes. These included AAVs 1, 2, 3, 4, 5, 6, 8, and 9. These samples were denatured and diluted to 4 times 10 to the 11th GC per mil for analysis. For all 8 serotypes, a minimum of 3 peaks can be seen, and in several serotypes, up to 5 peaks are readily quantifiable. Each serotype not only has its own unique series of peak, but its own PI values and peak ratios, showing that the method can be used as an identity assay. These data are quite interesting given the similarity between serotypes. While most of the AAVs exhibit a similar profile, there are certainly some intriguing serotypes to call out. First, AAV5 is the most acidic in the method, and AAV6 is the most basic. If we look at the table on the right, AAV1 and AAV6 are 99% identical. If we look at just those 2 serotypes, we can see the difference by IEF. AAV1 and AAV6 actually differ by only 6 amino acids shown here. I calculated the PI of each 6-mer to show you how much more basic the AAV6 peptide is, which explains our data and shows how powerful isoelectric focusing is. Both platforms provide comparable denatured AAV-IF data, which again means that these platforms together can support an AAV program from start to finish. The methods in the table below showed produce comparable percent peak area distributions for the same AAV sample. While the sample is diluted 10 times more for Simple Western, the profiles as well as the number of peaks come out quite close, showing that the information learned upstream in complex samples with Simple Western can be applied with later stage versions of the product. Using additional viral protein-specific antibodies allows us to identify which peaks in the complex denatured AAV profile come from which viral protein. Here we show an anti-VP1 detection shown in orange, in addition to VP123 detection on Simple Western shown in blue. Given that the separation of this AAV sample is nearly identical on the two systems, we should be able to use the peak ID information from our Simple Western data on the left to assign peak identities to the peaks in the Simple Western data, like this shown on the right. Knowing which peaks or which in a viral protein sample are important, for example, in a stress condition that appears to impact phospholipase activity of viral protein 1. One would expect changes specifically in the acidic region on the Maurice separation, which could be confirmed with a Simple Western run as needed. As we move on from the denatured method, I wanted to show one more example and then compare it to our intact method. So here we're comparing AAV8 and AAV9 with our denatured ICIF assay on Maurice. AAV8 and AAV9 share 93% identity, and unlike the comparison with AAV1 and 6, they overlay perfectly. Granted, they have different percent peak area distributions, but the PI values for each peak are essentially the same. If we now look at the intact method for the same two samples, we see that they have a different PI value from each other. Specifically, AAV8, shown on the top in blue, is slightly more acidic in its overall PI compared to AAV9 on the bottom shown in orange. So our intact or AAV particle characterization method is also an identity assay, and it can differentiate different serotypes. Now that we have an intact method, let's discuss a little bit of the importance of examining AAV particles. Measuring capsid content is quite important for AAVs, and it's a bit of a Goldilocks analogy, in that you want your AAV particles full, but not too full, and definitely not too empty. Along with aggregates, partially filled and empty capsids can increase the risk of immunotoxicity as well as reduce transduction. So they need to be monitored and minimized throughout the process and in the final product. There are several techniques currently in various stages of use to estimate this, listed here. In general, they take a long time, and most require modifying the sample in some way prior to analysis. Both Simple Western and Maurice are able to provide some estimation on capsid content. Let's now take a look. Keeping with our theme here, both platforms can again provide similar data for intact particle analysis. Shown here is the same pair of AAV9 full and empty AAVs, shown in blue and orange respectively. As you can see, the two provide similar data with the same overall benefits of each platform for each sample type. Let's now take a closer look at each assay. For our Simple Western intact AAV assay, we took a unique approach. First, as I just showed, we first separate the particles by IEF, detect them with the anti-VP123 antibody. Between these two AAV9 samples, we see about the same signal with albeit different peak shapes. Now here's the really cool part. In different capillaries with an anti-DNA antibody and the same samples, we're able to see a real difference between them. The anti-DNA antibody has a strong signal for the full AAV9, but a relatively weak signal for the nearly empty AAV9. These data can be quantified as shown below. Now again, this method has the ability to detect these sorts of sample differences in complex sample backgrounds, meaning there's no sample cleanup requirement. And then again, this is a very sensitive assay, so you can do this in your in-process or early stage materials where you don't have high concentrations. And a single cycle on a PEGI-SU IEF run can be completed in as little as 5 hours, giving you this estimation relatively quickly. Switching gears, I want to briefly discuss on how Maurice can be leveraged for intact capsid content analysis. For this, we are leveraging our absorbance as well as native fluorescence dual detection capability. In our absorbance channel, we expect to see both the DNA particle's protein signature as well as but albeit more strongly, a DNA signature coming from inside the AAV. However, in native fluorescence, which looks at tryptophan fluorescence at the same time as the absorbance, we should only see a protein signal as DNA doesn't fluoresce. To show that Maurice's dual wavelength detection helps elucidate empty full status of AAV samples, we analyzed a pair of commercially available empty full AAV9s with an intact method. First, shown in blue is the absorbance of an AAV9 77% full sample, and in black on the same trace is the native fluorescence. The two have high agreement for each peak. However, in contrast, if we look at the bottom electropherogram for an AAV9 empty sample, the absorbance is substantially lower. This makes sense given the fact that there should be very little DNA in that sample. These data can be quantified, and the native fluorescence can be used for normalization for any loading differences between the samples. When we quantified the absorbance and normalized it to the native fluorescence, as shown on the right, for the two samples, there's approximately a 5-fold difference, which is fairly close to what is reported by the manufacturer. So this shows that this method can be used for estimating the content of an AAV sample. We have also collaborated with Ultragenyx to look at their AAV8 program samples using the dual detection. Again, with the full samples as well as the intermediate samples, we see substantial absorbance and minimal absorbance for the empty AAV8s. When we look at the native fluorescence, all three are essentially the same. The data can be quantified on a per peak basis for both absorbance and native fluorescence as shown here. And these quantitative data can be used for a relative estimation of DNA content. We also looked at some stress samples with material from Ultragenyx that I'll show a little bit later. Stability studies verify that the storage conditions maintain the purity and potency of the product for a defined period so that the product administered to the patient is still capable of meeting the stability specifications. We have two platforms that are quite well suited for stability assays, our MFI and again our Maurice platform. Our intact ICIF method is stability indicating, and we believe it to be more sensitive than conventional stability assays. Shown on this slide are two examples of stability testing for AAV9. In the first test, we incubated AAV9 at 37 degrees C for 3, 7, or 14 days, and then ran them on Maurice compared to a control sample. In as few as 3 days, changes can be seen in the AAV9 particle. And certainly by 7 days, the AAV9 particle has started to significantly shift to a more acidic PI. By 14 days, the particle is shifted completely off from the basic original PI. Given that AAVs require low pH for activity through structural rearrangement, these data suggest that changes in AAV particle stability could be due to structural changes in the capsid. This method can also be used for formulation development. From the method, as can be seen from this series, the overall PI of the intact particle can be estimated to be approximately 6.6. Shown on the right is the same AAV9 particle sample, this time incubated at pH 10 for either 4 or 7 days. The change is much more substantial at 4 days compared to the temperature stress at 3 days, and at 7 days at pH 10, the AAV has changed as much as 2 weeks at 37 degrees C did. So the AAV is substantially impacted by storage in pH 10 buffer. This sort of information is available at 10 minutes per injection and requires very little sample, making the method ideal for both stability tests as well as formulation development. As the AAV sits longer in these stability tests, there is a higher propensity for AAV aggregates to form as well. Let's now take a look at how we can monitor AAV aggregation. Our microflow imaging platform, or MFI, is a great tool for AAV aggregation analysis and already has a significant install base in biopharma. MFI is able to support USP and FDA-mandated particle counting in biopharmaceuticals, as regulated, for example, by the USP 788 and 787. MFI is used for counting biopharma product aggregates like those found in monoclonal antibody solutions. The MFI platform features direct dynamic sample imaging for high statistical sizing and counting accuracy across 1 to 300 micron particle range. We have coupled the MFI with high-powered analytical software that is 21 CFR compliant, and for higher sample number, there's an automated option with the Bot 1. Voyager Therapeutics has leveraged MFI for AAV2 process development and published some of those data summarized here. In this work, they are looking at the ultrafiltration of AAV2 process samples and the influence of different ultrafiltration membranes. Shown in these two graphs are the MFI particle concentration data. On the y-axis is particle concentration as counts per mil, and on the x-axis is the size range from 1 to 2,000 microns. And in these data, we're comparing two different lots of the TMP membrane, as well as two different gauge pressures, either 6.5 or 20, shown in blue and orange respectively. The data show that using the lower gauge pressure, shown in blue, produces higher particle counts across the entire size range. So with a very fast analysis time of only 6 minutes per sample, the MFI is able to provide very valuable information about AAV aggregation during process. We've looked at both Maurice and MFI and how they can provide stability monitoring and are useful for process development and later stage characterization. As part of stability testing, a potency assay should be conducted to measure changes in potency as a result of storage. Potency is defined as the therapeutic activity of the drug product by the USP. And together with dose, potency defines the biological activity of each lot. USP Chapter 1047 indicates that bioassays and functional assays are typically to be used for potency assays. However, it also states that when the biological function of the expressed transgene exhibits a broad range of activities or only generates semi-quantitative results, the ELISA or other immunological or biochemical readouts can be used as a surrogate potency assay. Our SimplePlex and Simple Western platforms can be a strong supportive tool in surrogate potency assays for you. Let's take a quick look. There are two types of potency assays, and SimplePlex and Simple Western can support them both. First, there's something called a physical viral titer assay. Physical viral titer is used in dosing in preclinical settings and is typically done today with ddPCR or qPCR, STS-Page, or ELISA. There are challenges associated with each of these assays that can be overcome with Simple Western and SimplePlex. These include better reproducibility, better sensitivity, and better quantitation. The second potency assay is called the functional viral titer assay, or infectious viral titer. In this assay, typically cells are infected with a serial dilution of virus to determine what's called the TCID50. In this case, again, Simple Western may be more useful in measuring expression of the expressed transgene. I've already had the opportunity to show you some nice examples of our Simple Western AAV immunoassay, but here is how it can be used for a physical viral titer. Shown on the left is a titration of AAV2 sample of known concentration from 6 times 10 to the 10th down to 1.9 times 10 to the 9th GC per mil. And inset is the lowest concentration we tested to show that we still have really nice detection at the bottom end of the assay. On the right are the curves for each of the viral proteins individually, and then also in yellow as the total peak area. All peaks show good linearity across this range. And if you look at the lower 4 points of the assay, the R-squared values are almost all above 0.99. So if you have a known standard like the ATCC reference material, you can estimate unknown sample viral titer quickly with this assay. Simpleplex is an automated ELISA, and our instrument shown here, Ella, is well suited to replace any ELISA in your gene therapy analytics arsenal. Ella runs ELISA assays with improved sensitivity and dynamic range and ultra-high precision. The platform only requires 25 microliters of sample volume per well and completes in about an hour and a half. With Simpleplex, you can multiplex with confidence and without concern of cross-reactivity due to the geography of our proprietary microfluidic plates. These plates come in a variety of formats shown on the bottom right to provide sample input and analyte flexibility, supported by a broad assay menu. We even have an open cartridge format for those who want to take assay development into their own hands. Let's take a look at two viral physical titer assays that Simpleplex can support. If you are looking for a bit more of a closed-down assay with even more sensitivity and speed, then our Simpleplex AAV assay may be for you. We have partnered with ProGen to put their antibodies in our Simpleplex assay cartridge. Our Simpleplex platform solves key challenges associated with ELISAs, including much faster assay with minimal hands-on effort, better quantitation, and sensitivity. The Simpleplex ProGen ELISA can run 72 samples per run with a built-in standard curve. Shown here is an example of that built-in standard curve with a range between 10 to the 6th and 10 to the 10th capsids per mil. The standard curve and assay overall have excellent sensitivity and correlation to the ProGen AAV2 ELISA, which gives good confidence in the ability to accurately measure physical titer. Looking at some control material, shown in blue here is the ProGen lyophilized control, and in orange the ATCC reference material. And for both the high and low controls, we have excellent reproducibility, as well as good spike and recovery and dilutional linearity across the entire test range. Similarly, we have a fantastic Simpleplex assay for measuring P24, which is an HIV protein. It's also found on the surface of lentivirus. Quantification of P24 can be used to estimate lentivirus titer. Shown here is the built-in calibration curve with a limit of detection of 0.67 picograms per mil. The assay has strong performance with interassay CVs under 10.2% and intra-CVs under 7.1%, and shows good linearity and recovery in several sample types. Now, the assay performance shown here clearly doesn't spell out lentivirus work, but we know it's being used in that way. In fact, shown here is a pre-press article from GSK where they're using this very assay for lentivirus titer according to the manufacturer's instructions. We also have developed a simple Western assay to estimate lentivirus titer by measuring P24. We're still working on this, but thus far we have an 11-point two-fold titration curve for recombinant P24 in 100 microgram per mil HEK293 lysate. The assay right now has a dynamic range of approximately 2.5 log, with LOD and LOQ around 1 nanogram per mil. We intend to continue working on this assay and start testing some lentivirus samples soon. This assay has important benefits we see compared to the Simpleplex assay, which, as we covered, is great for its purpose. The Simple Western assay can be multiplexed with a target of interest and works well with cell lysates and being flexible to various lysis buffers. In addition, it is still quite sensitive with the LOD and LOQ being around 1 nanogram per mil. It's critical to understand not only the viral titer, but also the genomic titer, and we have an assay that can do both at the same time. For this assay I'm about to show, we purchased a series of AAV9s of known percent full. We normalized them according to the viral particle per mil from the manufacturer, and then analyzed them on Simple Western using an anti-DNA antibody, and then replex for total protein to quantify the AAV viral proteins. To start, shown here is the VP signature for each sample. You can see that even though we normalize these, the 5% is still a bit low by VP signal. We'll come back to that. Now, with an anti-DNA antibody, we can see increasing signal at a pretty high molecular weight. Next, let's look at our lane view with our built-in normalization to account for the differences in VP signal. Displayed on each lane is a blue dot. The 20 to 90% full samples all have VP signal within 10% of each other, while the 5% is about 60% lower for viral protein signal. Using all of these data, we can generate a correlation plot between the percent full for the manufacturer and the VP protein normalized DMA signal, where we see excellent agreement. Being very new, we still have more to do with this assay, including exploring other serotypes, as well as a little bit more optimization on the separation conditions. Lastly, let's take a brief look at an example of how Simple Western is being used for functional viral titer. Dystrophin is a large protein found in muscle cells where it connects actin filaments to the dystrophin-associated glycoprotein complex, or DGC, on the muscle cell membrane. In the absence of dystrophin, the DGC collapses, leading to muscular dystrophy. Shown here are data from a Nature Medicine paper where they're using Simple Western to detect dystrophin. What the authors show in this figure is that AAV6 Cas9 mediated excision of exon 51 in DMD delta 52 patient myoblast or cardiomyocytes restored dystrophin expression. The authors also went on to show that the restoration of dystrophin had downstream biological impacts. The bands here represent the DP427 form, as well as the shorter dystrophin isoform DP71. And the researchers used beta-actin as loading control. On the right, dystrophin levels are shown normalized to beta-actin expression as percentage of mean levels in control cells. So this is just one example of how Simple Western can be used to look at transgene expression following treatment with the gene therapy. In conclusion, Bio-Techne provides innovative, customizable solutions to meet your cell and gene therapy research and manufacturing needs. We touched on the fact that viral vectors are powerful and complex delivery tools for cell and gene therapy. And I have hopefully shown you today some great examples of how Protein Simple provides premier analytical instrumentation ideal for this space. With our Simple Western platform, we can supply you with a platform for early-stage gene therapy R&D and provide viral protein-specific AAV detection and complex samples. Maurice is ideal for formulation and late-stage characterization, with CESDS being ideal for AAV capsid ratio and purity analysis, and ICIF providing both identity and stability indicating assays for AAVs. Our SimplePlex and Simple Western platforms can both support viral particle titer for lentivirus as well as AAV. And lastly, MFI is an ideal image subvisible aggregate tool that's already been shown to be useful for AAV aggregation. Now with that, I'd like to thank you for your time and attention today, and I'm happy to answer any questions.
Okay, thanks, Chris. So the first question is, the isoelectric focusing data is interesting. Any comments on what the peaks are exactly for VP proteins? I had expected just 3 peaks, one corresponding to each.
Yeah, that's a good question. So, in addition to the viral proteins having their own intrinsic PI, there are post-translational modifications that can occur on each individual VP, and that certainly differs across serotypes, but we expect that some of the heterogeneity is due to that.
Okay, and you mentioned purity in a few slides. Can these systems detect contaminants in the process and things like host cell proteins?
Yeah, so I didn't actually touch on that too much today, but both Simple Western as well as SimplePlex can certainly run host cell protein or HCP assays. We have for SimplePlex a CHO cartridge with the Cygnus 3G material in it, and Simple Western we've demonstrated CHO and HEK and a couple of other HCPs.
Okay. For the Simple Western systems, can you comment on how the multiplexing is done Is it like strip and reprobing?
So you can multiplex on Simple Western through a variety of approaches. Certainly, you could put the two primary antibodies together and just go straight at it. We do have the replex, which is like strip and reprobe, with nearly 100% efficiency of removal in between. So you do have that option. We also, on a JEST, you can multiplex between colors or between chemiluminescence and fluorescence. You can also multiplex an immunoassay and a total protein assay, just to finish out the menu there.
Okay, and as a reminder to the audience, you can go ahead and type your questions in at any time in the question answer box on your screen. Okay, so can you comment on why the IEF peaks change between intact versus denatured AAV samples?
Yeah, so and this has been published, the viral proteins themselves, of course, have a isoelectric point, as all proteins do. But the capsid itself, when it's formed, also has its own PI, and that thing seems to be different in almost every example that we've looked at in terms of its PI versus the denatured proteins. So yeah, the capsid has its own PI, and that's important physiologically. It's related to how the AAV gets into cells and eventually opens up and delivers its material. So.
Okay, and the theoretical capsid ratio is 10 to 1 to 1 for VP proteins. From your data, can you comment on the measured ratio of 7 to 1 to 1?
Sure. So yeah, 10 to 1 to 1, VP 3 to 2 to 1 is the theoretical ratio of the 60-mer capsid. However, it varies between serotypes as well as even preps of the same serotype. So AAV2 has been shown a number of different ways to be roughly 7 to 1 to 1, like we showed today. AAV9, for example, is closer to 14 to 1 to 1. I showed an example of that where we measured it to be 11 to 1 to 1. So it definitely varies between the serotypes, but we can actually accurately measure it. That's what's important.
Okay, and can you comment on why you would want to measure AAVs in either denatured or intact forms? Why not always intact?
Yeah, that's a good question. So certainly the intact particle, as I showed, is quite interesting to follow in a stability test with the Maurice data that we looked at. And certainly if you're trying to characterize the intact particle and what's inside of it. The denatured method, you have the ability to look at stress changes to individual viral proteins. And as I showed through the comparison of the Simple Western and Maurice data, we know roughly which VPs are which in that separation. And so if there are massive changes on one side of that chromatogram, we know that there's a stress there, and that could be important for developing a more robust AAV where you, for example, modify some amino acids in VP1 to make it more stable.
Okay, well, I'm looking for some more questions, but doesn't look like we have any more questions. So thanks, Chris, and
Sure.
Thanks to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.